mention, there's a sign-in sheet, so if you've not signed in, please do so. That helps us justify the pizza and the food. So I'd really appreciate it if you could sign in. And so today we have Jon Mowers, who's going to talk to us about an interesting resource that he's been working on.
Yep. Thanks everybody for coming. As Marcy, and thanks Marcy for the introduction. This is a project that I started on my vacation and interview months during medical school with my little brother. And so this is actually the first time it's been presented to people. And I'm very excited about this web tool, and I think that it has a lot of potential to change the way we as researchers reference information. And so what I'd like from you guys as people seeing this presentation is to hopefully take home some points for yourself, maybe be able to use the resource moving forward to further your own research, as well as give me some feedback on how to use this resource. So the resource's name is PubMow. And the name of the talk is Modeling Biological Pathways by Redefining Referencing. And we'll be elaborating on how this web tool actually performs this function. So this tool is really born out of my PhD years, wishing that I had a tool like this. So I saw several problems within research that were occurring broadly. There was a failure, and I'll go into each of these more in depth. There was a failure of communication among researchers as we used different jargon to describe our work. A failure of narrative, and what I mean by narrative is packaging many experiments together to fit a consistent story. And also a failure of recall by each individual researcher to remember exactly which experiments were performed that define the foundations of a particular field. So with respect to communication, as I said, different researchers use different terms to describe the same phenomena. Potato, potato, solanum, tuberosum. Tomato, tomato, solanum, lycopersicum. These are all the same thing, but different people may say these things differently, and they all however, they all refer to the same thing. And how can we precisely describe what's happening when we're unclear about who the participants are? So part of this tool is addressing this problem. A failure of narrative, and as I mentioned, narrative being many experiments put together. And what's true about most narratives or stories you've heard outside of science? Well, one thing that is true about them is that stories are often untrue. And we've had a lot of problems with irreproducibility within science. In 2011, Bayer found inconsistencies, reported that they found inconsistencies in 2/3 of the work that they tried to replicate. In March 2012, Amgen found only 11% reproducibility of experiments that they were using to help further their work in the pharmaceutical field. And retractions have grown tenfold on the last decade, but are still only 0.2% of papers are retracted. Therefore, you have a very large discrepancy between the reproducibility across labs and the rate of retraction. And an example of a narrative that has been plagued with problems was the 1998 Lancet study that proposed a link between bowel disease, autism, and the MMR vaccine. And it took a very long time. However, in 2010, the Lancet formally retracted this paper only after many papers were published to the contrary. So reasons for this failure of narrative, well, with respect to the Lancet case, fraud is suspected. Then there's also misinterpretation of results, as well as poor statistical methods, failures in peer review, and also the pressure to publish or perish, or too many narratives being put out too rapidly, leading to I'm using this term in a somewhat pejorative, hypothesis-driven science. The idea that you, as a researcher, you have an idea, and then making the data fit this idea. So another problem we encounter is failure of recall. Just thinking, trying to remember exactly where in the literature we encountered these experiments. And some people are more prone to this than others. I have a series of brain scans here, varying activities. And here's I took a scan of Dr. Koenig, and it was very vibrant. And then there was one of mild cognitive impairment. And then there was, when I got myself scanned, I showed some pretty significant deficits, ventricular enlargement. So some people are more prone to this problem than others. So, to summarize the problems that PubMao was set out to address: failure of communication among researchers, failure of narrative, the idea of many experiments being packaged together, and the failure of recall by each individual researcher. And I set out, what allegories could I establish that really encapsulated this problem or defined this problem? And there are several ones that failed pretty miserably. We had Teos, found downtown, with respect to failure of communication, the menus in a foreign language, failure of narrative or product. They have this tasteless, fibrous meat matrix, which is not chicken, and failure of recall. I've never had a memorable meal there. Some of you, let's see, can we get the lights down up front, actually? Just on this side. For those of you who watch television through the 1990s, I don't know how many of you here actually are. We have Twin Peaks. I didn't understand it in the moment. Here's Log Lady down there. It had the worst conclusion possible, and I had no memory of how we got there. And finally, for 90s music buffs, there's the song Informer by Snow. It was kind of catchy, but what was he saying? He's saying, a licky boom boom down? I didn't know what that meant exactly. And it turns out it's a song about him being worried about snitches, and the narrative didn't make sense with this. It looks like an accountant, pretty much, being worried about being on the wrong side of the law. And very few remember that. So none of these analogies perfectly fit, but I did settle on Transformers, actually. I loved Transformers when I was a kid. They have many facets and components, and when you put them together in different ways, they can assume different functionalities. You see here we have the semi, here we have a launching combat pad of sorts, and then Optimus Prime in his full glory. So I thought of this very similar to an experiment. An experiment has different components, different facets, and when you alter them precisely, you come out with different functional states that you can observe, much like the Transformer. However, when you group a bunch of these experiments together and try and construct a narrative around those, sometimes it can have disastrous results. And for those of you who aren't familiar with the Transformers movie series, I'm going to say just don't waste your time. And here's Michael Bay, the person who drives this project, and says, I didn't even have to get this. It says, I am Michael Bay, destroyer of childhoods. I just found it randomly. I didn't even have to make that. I was pleasantly surprised. So how does this allegory carry through? Failure of communication. Energon, Allspark, Megacycle, Matrix of Leadership, what are these things? Failure of narrative, superficial characters, superficial themes, inconsistencies, plot holes. Wall Street Journal described it as a pile of glittering junk. And failure of recall. I have no idea what happened during these movies, despite seeing all of them. Was this a matter of amnesia? Did I actively suppress them? Or did my subconscious repress them because the plot lines were just that stupid? So how does PubMao address these different problems? Well, with respect to communication, as we described it, researchers use different terms to describe the same phenomenon, and it introduces an idea that many of you are familiar with, ontology, a shared vocabulary to characterize the types, properties, and interrelationships of concepts. Sorry. And so, to get us to all speak the same language, we need to draw from the same dictionary. And to do this, I thought I relied on the NCBI, the National Center for Bioinformatics, for my vocabulary. And we have many different categories of biological entities that we examine within in biology, and there are corresponding NCBI databases to them. There's the gene database. For transcripts, we use the nucleotide database filtered for mRNA. For proteins, there's a protein database. For small molecules, there are the PubChem databases. And then for processes and diseases, there's the medical subject headings database. And the last database that actually serves as a further degree of ontology is the homology database, which is actually describe each gene, transcript, and protein across all eukaryotic species with a single term for each type, all based on homology. So to find these within a regular PubMed browser, you would just click on PubMed up here, and you see it highlighted down here. It's one of the many NCBI databases. And gene, nucleotide, protein, PubChem, the medical subject headings, and homology are all found there. And so we rely on these databases to derive our terms. And we're going to be going through and doing an example in which we can see this in action as well. With respect to a failure of narrative, what PubMed gravitates towards and proposes is a system by which individuals are allowed to publish and reference individual experiments as opposed to whole works. This allows individuals to characterize work previously done, found usually within a single figure and a single letter within that figure, as well as publish experiments that counter previously published work without having to commit to an entire counter-narrative. It's very difficult for people to get funded to publish a work that is described as negative or contrarian frequently. It also serves as an annals of null results, or for an individual researcher, it could be a lab notebook. As far as failure of recall, this one's a little bit simpler. We currently cite things in a bibliography, as you see kind of down here below. And PubMed promotes publishing individual experiments, like I described previously, and you have to designate the journal figure number and letter, and it's stored and easily retrieved. So this is what a paper I published in last December looks like when rendered by our system. I figured I'd start with kind of an end product. You have a biological pathway in which many of these different biological entities are interacting, and a directionality to it, in which you have arrows suggesting some increasing either activity or transcription, and red T's indicating inhibition. And so you can see that there are many experiments here within this paper that related these different entities, and so, between two entities, there were many experiments that describe that relationship as well. So how do we get there? We get there by describing individual relationships one by one, essentially. And what you can see is we start out with a cause, which is normally the independent variable, an action, increases, decreases, it doesn't change, and then an effect, which it would be the dependent variable, and then specify precisely where it's found. So to do an example of this, I relied on a pathway that a lot of people have encountered previously, adrenergic signaling. So here you can see epinephrine signals through beta adrenergic receptor to stimulate these G proteins. You've got a heterotrimeric G protein. G alpha dissociates from beta and gamma, goes on to stimulate adenylate cyclase, which produces cyclic AMP, which then goes on to stimulate protein kinase A. So the very first component of that would be epinephrine leading to stimulation of an adrenoreceptor beta 1. So here you can see a very simple experiment. You have increases in epinephrine from 0 to 10 lead to increases in adrenoreceptor beta 1 activity. And it's a statistically significant change. So how do we encode this? Well, we change our independent variable, or our cause. It would be small molecule, put in epinephrine, then click on our action, say that it increases based on the fact that this is a statistically significant increase, and then, indicate that our dependent variable, the adrenoceptor beta 1, is increased, and specify where in a particular paper that is found. For simplicity's sake, I'm just going to use this figure. Okay. One thing that you can also do is, in addition to citing it from, if this were coming from a paper, you can also choose that file and upload it yourself so that individual experiments you do can be uploaded. So, with respect to this system, I was hoping to then go through quickly and show how this would actually be done. So we, here at pubmao.org, in the add relationship, we said small molecule, epinephrine, and then it sends over a query to the to PubChem and asks for a list back of things that fit epinephrine, and then the NCBI, hopefully this research will pop up pretty quickly. Come on. We'll fill this in while that's getting piped through. The reason the small molecule takes longer than others is because it actually goes through two different search engines. First, it goes through PubChem Substance, then it goes through PubChem Compound. So, let's see. Oh, did I spell it wrong? Yeah. Epinephrine. There we go. So I spelled it wrong the first time, sorry about that. Since we're making we have our table number and figure letter. And then when we save this, we're able to see that a connection is made between epinephrine and the adrenergic receptor. And a summary down at the bottom describes small molecule epinephrine increases the adrenoreceptor beta 1 based on this PubMed ID, this figure. And this is, other individuals can come in and edit this if they'd like, so that it is an interactive database. So take one of those, and then we'll just take the next step, which was the adrenoreceptor. and that connected to the G alpha, which is known as GNAS. And if we were to assume it was the next figure in a paper, the figure 1b perhaps. And when you save it, then you can see that a system is emerging in which you have epinephrine leading to adrenergic receptor, leading to G alpha S. And
Is increases a good word for that?
so that's a
As
good
opposed to
question.
activates?
Yeah, so activates would be more precise in this particular instance. I agree. And I think that there's a lot of room for post-translational modifications or precision in language. We were trying to make the most broadly applicable term for this, for biological processes. Activates could be that, however,
Well,
it could increase phosphorylation. It could increase
That's going to be
one
the
of many different
stickler,
things.
because some things increase the quantity
Uh-huh.
and other things increase activity, for instance.
Sure. No, it's a very good point, and there's room in the description to further elaborate on the type of relationship between the two entities that you're observing. But the terms that we settled on were just as broad as we possibly could. So.
Another question would be, it seems like the way you input information into your system is like relationship centric, and in many cases that if you publish a paper, your single figure might tell a multiple relationship, and in this way you might need to do multiple uploads or multiple reference for a single figure, but tells multiple things at the same time. Probably that would be a little laborious, right?
Yeah, it certainly is, and in the paper that I published, took 4 years of my work to generate, but it took about 2 hours to upload around 70 relationships.
Okay.
And so, yes, I think there is a little bit of work and to be done, and in the future we'd like natural language processing or something along those lines to be able to go in to individual figures, individual parts of individual figures, perhaps even interpret the graph and automatically upload these relationships. But I think we're not at that point just yet. And so for the time, yes, it is a little bit laborious to upload every relationship within within an individual paper, but that's also hopefully where crowdsourcing will come into play.
Yeah. Silly question maybe, but I mean, a batch upload, for a given set of fields seems like a pretty good idea for situations like you're describing.
So scaffolding is how me and my little brother have been discussing it. There are a bunch of pathways, including KEGG pathways, wiki pathways, that the Reactome, that have several well-categorized versions of these pathways. And so I'm hoping in the future to scaffold the site such that you can see pathways that have been established before and hopefully are well vetted, and then have people go in and put in the precise point in the research, in the literature, that those relationships are found.
That's not what he was asking.
Oh.
I'm saying if you have a single paper and you have 10 figures, and on each figure there's 5 different relationships for those 50 different relationships, can I upload an Excel file with a set of predefined columns?
So
then,
you could. I think what you would miss out on would be the ability to search through the NCBI. So the relationships there would be the relationships that you had predefined, and you can freeform enter terms. What this relying on the NCBI databases allows you to do is narrow your focus to, yes, this is the term that I wanted to use. And so I think, yes, you certainly could. But then how it links to other people's research might be hindered because you're not using the same vocabulary as other individuals. But
you could probably get around that. You can have like an input wizard where if you put in 50 relationships in an Excel that it says, are these the terms, just automatically does the searches for each field.
Sure, gets you through and to validate it.
Yep.
Yeah, so you wouldn't be able to upload it directly as the Excel, but you'd still have to go through and say, is this the term? Is this the term? Is this the term? And yeah, we certainly could make a tool like that.
But I kind of fail to see how that solves the problem of communication. So I think that, you just created yet another network, and what you entered is subject to interpretation as much as any other scientific result. I mean, I agree that it is a problem, but I just don't see how that, I think, in a way, I can argue that it compounds the problem.
I mean, so I think it's an imperfect system, but we believe it to be perfecting. You have, like with respect to compounds, we link everything back to PC compound, which is supposedly one of the better annotated ways of describing any molecule. PubChem substance is a little bit more broad. Many things, potato can both exist within PubChem substance, but they will only be represented by one molecule within a PubChem compound. And so that's the hope is to get to, at least when one person says potato, to at least get them to the right place. I don't think it doesn't solve the problem, certainly. And the NCBI as a foundation for all of the vocabulary is perhaps a bit of an overreach, but I couldn't think of a better system that integrated well with our search capabilities, and so that's why it was used. Yep.
Is there any way to remove like a blatantly obvious error that shouldn't be there? Like, for example, if somebody came up with, if somebody decided to just do something that blatantly contradicts the current literature, like say lead somehow increases neurological activity in a certain receptor, but you should do the opposite. You should do the opposite. Is there any way to remove that arrow? Or
So we wouldn't say there's a way to, if that arrow is linked to a specific piece of data, we would prefer that if it is in, and that piece of data is either published or uploaded by an individual, we would prefer that it accurately describes what everybody else is seeing on the paper. However, if two results come in conflict, then you can actually you can actually get both an arrow and a T going between the same two biological entities. And then when you click on one, you see the evidence for if it's one entity increases another, you see the evidence for that relationship. And if you click on the red arrow, red T, you would see the data that relates to this entity decreases this other entity. Does that make sense? Okay. So, any other questions?
Actually, just one. I don't know enough about this, but if you're using figures from published works, are there any kind of, it's a silly question probably, but copyright issues? Because generally you hand over your...
You can't upload the precise figure. So the relationship between these two, the data itself, I don't believe to be formally copywritten.
Data can't be copyrighted.
And so...
figures themselves.
But if you were to upload somebody else's figure or a figure from another paper, that would not, we wouldn't consider that good. But you can link directly to it, and you can link directly to the PubMed ID, and when you click on that PubMed ID in the system, then it takes you directly to it.
You stop people from uploading figures.
No, I can't say that I'll stop people from uploading figures at this point, but perhaps we can get a marshal of sorts. Let's see. So what we're showing is that you can take the same system and through simple causal relationships recapitulate it. And as you mentioned, there could be controversial relationships, such as between thimerosal and autistic disorder, and you can capture both pieces of information based on by including both errors.
If you get a number of papers that build up the red, will that get thicker and thicker and thicker?
So yeah, number of references will in the future be related to arrow size or opacity. We haven't decided which one is best. So in summary, we are modeling biological pathways by redefining referencing to the individual experiment, as opposed to referencing an entire paper to describe what's going on between different biological entities. So PubMed is a publicly curated site that allows users to electronically publish individual experiments as well as reference individual experiments previously published and then automatically generate an interactive model. Problems addressed by PubMed, we think that it improves communication by getting people to use the same vocabulary based on the NCBI. With respect to problems of narrative, we allow individuals to record individual experiments as opposed to require that they produce a full story. And in terms of recall, you can fairly easily store and share where this information is found. Future plans, I mentioned scaffolding biological pathways, building user community, perhaps through Rackham or the MSTP. If anybody here would like access, I'm glad to give it to them. Plan for the future is if you have an EDU email, you can get on. Improve visual representation, including size, opacity of nodes and arrows, as well as way down the line, try and farm the literature for these different relationships. Because if we don't intervene at this level, I'm afraid that this guy wins. And nobody wants Michael Beta. And so acknowledgements, I've been talking with Dr. Koenig, Athey, and Belize, my little brothers here. This is us in Yosemite. We made a snowman. I want to thank him very much for being a part of this project. And welcome any other questions.
How serious were you about the lab notebook usage?
I really like that idea.
So you're ready to do all the things about access control, secure backup, off-site backup. If you crash somebody's data, you're taking on an awful lot.
Yeah, that's true. That's true. And if it gets that large, I'd be happy to have that problem.
Oh, 2 questions.
Yep.
First, you just say, so your topic is redefining reference. I would like to see, is there any way that I can reference the interaction in your project instead of reference a single paper to express the idea in the actual paper?
I mean, potentially you could. Each relationship has its own ID within our system.
That's what I want to see.
Yeah. So let's see. Sorry. So is this causal relationship 201?
Okay.
And so, like, if it were a viable system, if it is a viable system, then you could reference that individual experiment or series of experiments, causal relationship 201 through 208, those types of things.
Okay. Second question would be, so if this system is viable and we get more and more data getting into that, as he asked that we might have controversial results in a certain relationship. I don't know whether you have any way to give the weights to different interactions, especially like nowadays information retrieval have a lot of algorithms to processing this kind of graph-based network weighting to give some certain kind of pseudo confidence for some published data or interactions.
So establishing a metric for weighting
Yes.
confidence for relationships,
Yes.
I think is a question that remains for us. The simplest and most likely simplistic version of that would just be how many figures does it show up in? If this relationship is recorded by individuals 10 times, then it gets a certain weighting, maybe a logarithmic-based weighting to it. But in terms of improving on that weighting, that would be something we're interested in too.
Okay. So because you just mentioned in your slides that there's a consistent gap between like the controversial discussion about certain interactions and the retreat of the paper. If there's no weighting or assessment system in this kind of interactions system, it's hard to see how it can help this kind of issue.
I agree, and it definitely. So we use Cytoscape Web as the way to. That's how the visual representation is done, and there we've looked through the different visualization documentation there, and there certainly are ways to change the weighting, and it'll be fairly simple to do, and something to look forward to for us.
If you want people to try this, you're going to have to get some help.
Yeah.
Documentation.
Yeah.
I took a look and it wasn't clear to me. I could make guesses from poking around a bit, but
sure.
What are the lists of acceptable terms for relationships? That we laid out.
Yeah. So my idea for that currently is to take these exact graphs, and then when you scroll over or tab into a field for this graph to pop up, such that people can, and there will be help documentation outside of that.
Yeah, okay.
But in terms of getting people started, to just have this graph along the side, so people are like, oh, the independent variable goes here.
You need the documentation outside of that strongly, because poking around and trying to figure out what's what is not a substitute or a tutorial to go along with it.
That's a good idea too. Yeah, tutorial certainly.
Sometimes contradictory experimental results are both correct, but it's the experimental difference, different cell line organisms. So does that get entered into this somewhere?
Let's see, cells, a lot of cell lines are accounted for in medical subject headings within the NCBI. As far as you can designate different environments as well. The different environments that are available to designate are in vivo, ex vivo, in cell /tissue culture, in vitro, and then in silico or pyro if you're real old school. And so let's see. And so there are ways to distinguish between these as well as abilities to
Like mouse strain, that can have a huge effect.
repeat that.
Strain of a mouse.
Oh yeah, sure, certainly. And those bits of information would likely fall in the description. You can specify if you wanted to look at a particular protein within a particular species, you can certainly specify that as well. But as you were saying, Val versus black sticks can behave very differently, and therefore it'd be nice to have that information in there as well. But at its root, we're trying to get at least some information in there, enough to make it a system that's worth using. I think the different advanced features will probably come as the system if the system gains traction.
So I would like to make a supplement to that question. So other than cell line and experiment conditions, also you might have mutated proteins, mutated sequences, and for small molecular binding, I say that I have a radio-labeled small molecule and normal small molecules. So far, I cannot tell that your system right now can handle that. And actually, when we are doing some natural language processing project, we also have the problem when we try to normalize this term, we cannot find a good way to express them in the database. So do you have any idea about this problem?
Yeah, so one thing that I'd like to do with that is have advanced feature for you mentioned like I think you mentioned phosphorylation as well?
Probably.
Or amino acid changes. So,
yes.
So mutations.
Yes.
That's what so different alterations. There'd be an advanced feature for each, if you used a special, each gene transcript protein, you could have an advanced feature that says mutated at this point, phosphorylated at this point, acetylated at this point. Those are the types of things that we're considering adding on. So.
Is there any way to distinguish between different types of, different organisms that you're in vivo for? I mean, different molecules cause radically different things. To use a simple example, if you have dioxygen in one species, if say humans, it isn't going to affect most of your tissues, but if you have it in anaerobic bacteria, it'll destroy it because of the ROS generated, even if they have homologs. So, if you say the dioxygen destroys this homology in this bacterium, is there any way to say, but it doesn't affect it in humans because it doesn't affect humans?
Let's see. So the way that this is set up, presumably you'd have a figure and a letter related to each of those relationships and in the different species. In terms of the larger categorization, I do foresee it appearing like select a species and then look within a particular cell within that within that species. That's how I think a good way of partitioning it off would be, because different cells also within the same species behave differently. And so that's the simplest way that I've conceived of partitioning it. And a lot of databases partition things by species as well. And then using medical subject headings to describe them. Yeah?
So I think in terms of, that sounds like it could get complex and capture a lot of sort of complex relationships. In terms of narrative, how would you see, imagine it's 5 years down the road and everyone's populated their data into the database. How would, I don't know anything about, your research. How do I get a narrative view of what you've done?
So you would be able to sort for papers that I've done and see the relationships that be able to select those relationships and import them exclusively. So,
what do I see that helps me make a narrative out of those relationships as opposed to just a sort of the earlier question, just seeing a network of things? How do I turn it into a story, or what's helping me understand more about it?
That's what the normal paper processing thing is for. We don't view this as replacing it, but we view it as a supplement to it, so that there certainly needs to be an in-depth discussion of what's going on. However, we currently don't see any way for people to publish small bits of data very effectively, and this is one way of approaching that problem.
When you say small bits of data, so far, to understand correctly, that so far you're only referring to the experiments published in peer-reviewed journals. Is that correct?
Yep, or things that individuals have uploaded themselves.
Okay, well, that's a big difference, right? So how is that sort of, if it's not peer-reviewed, okay, well, you rightfully said that peer-reviewed, publications have issues and, named some of the reasons that led to this data being retracted. If you open it up for anybody uploading anything, how is that going to affect the overall quality of the data, and how can you, as a researcher, trust that data that somebody just uploaded? Maybe I don't know the first thing about making these experiments.
No, I think that's a valid point, and I truthfully don't know the answer to that. I do know that an individual, a Harvard biologist, submitted to 300 papers an entirely concocted project with entirely concocted results, with horrible methodology, and was accepted in 150 of them. Peer-reviewed journals. 1998, there was an individual who sent out to, for I think it was BMJ, British Medical Journal, sent out 8 glaring errors in methodology as well as interpretation, and to 200 of their regular reviewers, and found nobody actually caught all 8, and the average was less than 2 for peer reviewers. And so I think that peer review certainly has a place, but I don't think that it's perfect either. I think that if you do upload something that looks terrible, then people can cite multiple experiments that show that it's terrible, and then you said your piece, they said theirs.
Sure, but this is complicated, right? So arguably, Wikipedia, everybody is using it, but it cannot be cited as a sort of, rigorous source of information, precisely because, I can go and, delete what I don't like and replace it with something which is absolutely incorrect. Agreed. But peer review is not perfect. Nobody is saying it is. You're absolutely right. But these things are complicated. Who's to be the judge?
Well, so if you're uploading a method with every data point that you project or substantiate, there's a peer review process has no better insight into what your method was than what you send. You send them a method. No one's looking over your shoulder. So the access to the peer reviewers and the access to the public, it seems to me at least, is identical in either case, except the difference is here, anybody can weigh in when they see an experiment they disagree with. Instead of waiting for it to be published or being one of the 6 people who reviewed the paper or 3 people, you don't have to wait for it to be republished and then send a letter to the editor saying, I think you should retract this. You could comment on it directly. So it seems like, if anything, this is a more transparent peer review system. It's solving some of the problems of peer review being limited to 2 or 3 people who have other things to do and want to get a paper off their desk, instead of these are people who actually care about the actual experimental interactions, right? So it seems like, if anything, it doesn't have to be cited, but you get exactly the same method that you would get in a peer-reviewed publication, unless people treat it less seriously, unless people just lie when they're uploading to this, but don't lie when they submit to a science or nature or something like that.
So yeah, and I think I'm of that mind as well, but in terms of how it'll actually play out, that's very difficult to predict. But
would there be a way to create a filter on it so that if someone really just wanted to see peer-reviewed interactions, they could just limit it to that?
sure, if it would have to have a PubMed ID associated with it, and then you could filter out things that don't have a PubMed ID.
Any other questions?
Okay, well, thank you guys so much for coming out.